The natural killer cells, they are not always cytotoxic. Okay? Natural killer cell cytotoxicity begins at sites of viral infections. Where there is infection, the natural killer cells would become cytotoxic or killers only in that tissue. Okay? But not always. They are not always cytotoxic. And the killing mechanism is not haphazard, just like somebody would grab a gun and start shooting it random. Okay? This is a very, calculated attack by the natural killer cells. So what makes it very organized and calculated? There are different mechanisms. For instance, the natural killer cell has to make a contact with another cell, with a cell that it is going to kill. So it's going to investigate it very deeply whether this cell has got some problem and this should be cleared or killed or not. It's a smart cell, not like us who would just kill anybody without knowing whether he is a culprit or not. So one thing is that the natural killer cells would make contact, make a synapse, and exchange information with the cell. It's not going to kill all the cells. The bystanders, there is no collateral damage. This term that human beings have devised when the military, for instance, is conducting some practices against the terrorists and civilians are killed. That is known as collateral damage. It's very easy to say that this is collateral damage. Okay? So the natural killer cells are smarter than human beings in that they do not do collateral damage. The bystanders or the healthy cells are spared. And it gets this information when it gets closer to another cell. Similarly, engagement involves several receptors and decision to kill is democratic, When it comes closer to a cell for inspection, for instance, and the synapse is made, just like here that you see, the natural killer cell is going to make a synapse and exchange this information with the help of different receptors that are present there on the natural killer cells and on the target cell. Okay? So in the synapse, all these receptors would be concentrated, and if the decision is to kill, that decision would be carried out with activating receptors, dominance of the activating receptors. And if there is dominance of the inhibitory receptors, the decision would be not to kill. Okay? There are inhibitory receptors that protect the cells because it's a natural killer cells. It's in the tissues there. In the tissues, not all the cells are infected. Okay? Many of the cells, they are not infected, they are not diseased, they do not have any problem. So the natural killer cell has to spare these cells. It moves from cell to cell, and it makes such kind of contacts with the cells, and there are receptors that are engaged. Some of the receptors are pro-killing receptors, pro-cytotoxicity receptor. Some of the receptors are inhibitory receptors, as shown here. So if there are more inhibitory receptors, in less dominance of the activating or cytotoxicity receptors, the cell will not be killed. So this is democracy actually, you know. If more of one type of receptors are there, engaged, it's not going to kill. So this killing mechanism is very organized and very calculated in the case of natural killer cells. Okay. Now you see this image here. This is a cell, and this red one that you see here is a virus. This virus comes here and infects this particular cell. So we are talking about the virus infected tissues, where the topic is natural killer cell cytotoxicity begins at the sites of viral infections. Okay? And I told you that natural killer cells are not always cytotoxic. They have to become cytotoxic or killers where there is viral infections, because it has to kill the cells that are infected with viruses. So in virus infected tissues, just like you see here, this is a virus. It has entered this particular cell. What is the cellular response now? What is the cell going to do when it is infected with virus? We already have studied this topic in detail. The virus, when a cell is infected with a virus, the virus infects cells and triggers the interferon response. The virus's response is to produce interferons when there is entry of the virus into the cell. Okay? So what is the virus doing here? It is going to produce interferons, interferon alpha or interferon beta. It means the cells in the infected tissues would produce these type 1 interferons. Alpha and beta interferons are type 1 interferons. Okay? Now in the tissues there are natural killer cells. These cells, when infected, in its RNA that you see here inside the cells, is producing these interferons now. So the interferons, they are going to influence the natural killer cells in the tissues. They are also in the tissues. I told you natural killer cells are in the tissues and in the blood. Okay? So in the tissues, the natural killer cells would come in contact with interferons produced by this cell. Okay? And what kind of natural killer cells are in the tissues? These are the CD56 bright cells. CD56 natural killer cells, bright natural killer cells, are inside the tissues. They are resident of the tissues. So these are the CD56 bright natural killer cells in the tissues, and these are interferon alpha and beta secreted by this cell, infected cell. Now they are going to bind to their receptors on the natural killer cells. They have receptors for them. Okay? So what will happen? There would be replication of this natural killer cells, or proliferation of these natural killer cells inside this tissue, as you see here. Type 1 interferon drives proliferation of the natural killer cells. There would be a cohort of immature cells formed in the tissue. Okay? So the natural killer cells would multiply in number as a result of this information. Interferon alpha and beta is the message for the proliferation of the natural killer cells. Now it would proliferate or replicate inside the tissues to give rise to a cohort of immature natural killer cells. Now these natural killer cells further undergo differentiation as a result of interaction with these type 1 interferons. Okay? Type 1 interferon drives differentiation of the natural killer cells into cytotoxic effector cells. What are the cytotoxic effector cells? These are the CD56 dim cells. So here you see that these are the CD56 bright cell, and as a result of interaction with interferons secreted by the infected cells, they proliferated and then they differentiated into CD56 dim effector cells. These are effector cells. Okay? Effector cell is a cell that is going to do its job now. The job intended to do here is to become cytotoxic or killer. Okay? This is the message for these cells to become killers. Okay? So these are now differentiated cytotoxic CD56 dim natural killer cells. Okay? And this cell that you see here, when it is differentiated, now this cell would come here in contact with the cells of the tissue. This is the tissue cells, and it would make a synapse I have given here, and in this synapse it's going to tighten the grip on this particular cell with the help of different receptors, just like CR3 receptors, LFA1 receptors. You have studied these receptors in the case of neutrophils. Okay? In case of natural killer cells also, these receptors are used here to tighten the grip on the cell. Okay? Now what happens here, that when the natural killer cells make a synapse to contact a cell, there are some changes that would take place in the natural killer cell. You know about this cell, that this is not just a cell where you see a round nucleus floating here and there. The cell is a very well organized structure, and there is a network of filaments inside the cell in which all these different organelles are embedded. So organelles are not freely moving in the cytoplasm. They have to be fixed at one place. So these are different microfilaments that are making the cytoskeleton of the cells. One is your body skeleton, which is made of bones. One is the cytoskeleton, the skeleton of your cells, which is made of different filaments, including these actin filaments. So what happens when the synapse or contact is made here by the natural killer cells with the host cell, that there would be reorganization of the actin microfilaments to align it with MTOC. This is organizing center of the microtubules organizing center, and the Golgi bodies for targeted delivery of the enzymes. The natural killer cells is filled with these granules, which have different kind of hydrolytic enzymes. It has different kinds of proteoglycans, or different kind of glycoproteins that are very toxic to cells. But these are inside the natural killer cell. Now when it has come in contact with this cell, it cannot simply just deliver these toxic products in the surrounding. Because if it delivers it in the surrounding, other cells, the bystanders or uninfected cells would also be killed. Okay? So for that purpose, the microtubules or the actin microfilaments, they reorganize inside the cell and they would make channels towards this synapse here so that the granules, when comes here in contact with the membrane of the natural killer cells, and fuse with the membrane, those toxic products are all poured into the synapse. Okay? And the synapse would deliver those products to the target cell, to which the natural killer cell is attached. What are those enzymes? And proteoglycans and other glycoproteins are going to do? They are going to induce apoptosis or programmed cell death upon this cell. Okay? It comes in contact with the products of the granules or the products of the natural killer cells. This cell is going to die naturally. Okay? It's going to die by programmed cell death. Where would the dead cell go? The dead cell would be taken up by macrophages. The macrophages would just dispose of all the debris that are formed here as a result of apoptosis or programmed cell death.